Dear all, thank you very much for participating to this webinar. As you know, this is part of a series of webinars organized by Work Package 4 of the ERICA project, that is the joint action for ERN in order to stimulate research. As you know, this is an opportunity for not only ERN healthcare provider, but all the people that are interested in engaging into clinical trial for patient. This is an important aspect in order to provide to the patient the opportunity to be treated according to their specific needs. Today we will go exactly into the aspect connected to the patient-reported outcome. And the title of today is, in fact, Introduction on Patient-Reported Outcome and Consideration Before Including Them in a Clinical Trial. Today it will be presented a study case of the ERN-EuroBloodNet, and as a speaker we will have Céline Desvignes-Gleizes from Mapi Research Trust, Andreas Glenthøj, it's a hematologist. The main objectives of this webinar will be to give an overview on patient-reported outcome measure and patient-reported experience measure in the clinical research. And the topic that will be treated are: what are PROMs and PREMs? What do I need to know about PROMs and PREMs before including them in a clinical trial? At what point on the preparation of a clinical trial do I need to think about PROMs and PREMs? And where I can find adequate PROMs and PREMs, and who can I address to have more information about PROMs and PREMs? Then we will have a case study issue from Andreas Glenthøj, that is, as I was mentioning before, a hematologist clinician that will explain the experience of using PROMs in academic clinical trial. Thank you very much for your participation, and I hope that you will really enjoy this webinar.
So hi everyone, I'm Céline Desvignes-Gleizes. I'm very happy to be there today with my colleagues and to introduce you the work that we are doing at the Work Package 3, the Patient-Centered Research in Rare Diseases program within ERICA project. And today we are going to talk about PROMs and PREMs and give you an introduction on different topics on this very interesting tool that could be useful for you in your research or your clinical practice. Today I'm not on my own. I'm with Andrea, who will introduce himself, but he will provide you with the perspective of a clinician and how to use PROMs in his own practice. So today, a learning objective of this webinar is really for you to give you a sense of the impact of PROMs and PREMs in the preparatory phase of a clinical trial. And we'll address 4 questions. What are PROMs and PREMs first? What do I need to know before including them in a trial? At what point of the preparation of the trial I need to think about PROMs and PREMs and if I include them or not in my trial? And where I can find suitable PROMs and PREMs, particularly in rare disease, because you know that it's a challenge, and who I can address to have more information on those measures. And then you will hear feedback, so both from a clinician and a patient advocate, from Andrea and Dor, on how their experience of PROMs use. So let's get started. So first, what are we talking about? I think it's important to start with the definition. And I'm pretty sure that if you ask many people in this audience their definition of a patient-reported outcome measure, I'm not sure we'll get all the same definition. But here, us at Mapi Research Trust, we are referring to the FDA definition released in their guidelines on patient-reported outcome in 29, and we rely on this definition. So it's a measurement based on a report that comes directly from the patient, and I think that's a very important statement, that it comes directly from the patient. About the status of the patient health without amendment or interpretation of the patient's response by a clinician or anyone else. We'll see that it makes also a difference with other type of measures. But the PRO can be measured either self-reported or can be interview provided, depending on the patient health status. So this definition is what we will rely on for the coming slides. Just to give you a little bit of maybe more a background, just to include the PROMs within the clinical outcome assessment measure family. And here we're just going to see the 4 types of what we call COA. This way, I think it could also help you to see what is so special about PROMs. So as we have just seen, a PROMs is a patient-reported outcome reporting patient health from the perspective of the patient. The second one is the observer-reported outcome measures, or an OBSROM. So here it's the same to reflect the patient health, but through the perspective of either the partner, could be sibling, could be a teacher, so someone that is not part of the healthcare staff, because this is the third type of clinical outcome assessment, the clinician's reported outcome. The same here, we are reporting patient perspective, but assessed by a clinician. So it could be, let's say, the healthcare staff. So it could be nurse, clinician, or someone that is trained for medical care. The last one is what we call the PERFO, a performance reported outcome. It's basically a task that we ask patient to perform, like stand up, walking 3 meters, and go back to seat. So those are the 4 type of outcomes that can be used and reflect patient health status. And here today we're going to focus on the left hand, the patient reported outcome. Depending on the population, particularly the pediatric population, it's worth using also OBSRO for obvious reason, but usually we can use PROMs or OBSRO depending on your population of interest. So just here, the 2 or 3 next slides are more about what is concretely a questionnaire, what should be displayed in a questionnaire in this PROMs structure. So first, usually, so here is just also to get some vocabulary around PROMs. So you should have instructions informing the patient what are we talking about, what is expected from him or her. Then we'll have the item, which is the same word for question. And this contains the concept, meaning what we are supposed to measure. Here, we are supposed to measure symptoms like itch, sore, or if it's painful or stinging. And also the recall period. The recall period is a very important notion where we ask the patient to which period of time they need to refer to. So here it's the last week. You have response option. Here we have 4 response option, the number of response option, and sometimes on some questionnaire, you also have directly the item score. All those items are gathered according to a set of what we call domains, which are concepts that we measure. So here, for example, symptoms and feeling, there are 2 items measuring these domains. And those domains, it's what we call the domains coverage of the questionnaire, will help you to see what are the domains that are assessed, and if they fit or not the actual impact of the disease in the patient's life. Here, if you know that the disease, it really impacts leisure or daily activity or work, maybe this questionnaire could fit your needs in terms of concept coverage. Then a notion on what it means to develop a PRO. because now, depending also on the context of use of your questionnaire, it's important to have in mind what is at stake when we develop a questionnaire, and then should we develop one questionnaire or should we adapt an existing one. So here, the same here, it's the PRO development according to the FDA guidance. So it's pretty exhaustive and obviously could be adapted to your need. But the first round is the item generation based on, let's say, 3 type of sources. The first one is a literature review on what is already existing in terms of questionnaires, but also to just give you a better sense of the impact of the disease. So to better understand the disease itself and how it impacts quality of life of your patients. The two other type of sources are interviews. Patient interviews on one hand and clinician interviews on the other hand. Those two, the patient interview is really critical, particularly if you develop a drug. It's really very critical to, at some point, get the feedback of the patient just to be sure that the items that you're going to generate actually reflect how disease impact patient's life. So after this first step, you will end up with a list of preliminary items based on a conceptual model, meaning that you have highlighted the concepts that are the most important for the patient. And items reflect this concept. The second phase is to test this preliminary version by first checking that the patient understands all items in instructions, the response options, all the different aspects of the structure of the questionnaire. Then we'll apply a set of statistical analyses, including the definition of a scoring, because we'll see that the scoring is very important in addressing some technical consideration. And then after modifying some items, maybe deleting some items that could be redundant, you end up with a final scored questionnaire. The final step is the measurement properties. So to ensure that the questionnaire is reliable, validate, and responsive to change over time. So with these three aspects, statistical aspects, you need to ensure that the questionnaire you developed is an effective statistic tool. Last but not least, you can see that it's time consuming to develop a questionnaire. It takes a lot of time, those resources, and money. So we also decided to present you PREMs, which are a bit different than PROMs, as you can see here. So here the objective of a PREMs is to measure the patient perception of their experience, the quality of care once receiving care. So they are different from PROMs because they are not really assessing the impact of assessing the impact of the process of care on the patient experience and not the outcome of the care themselves. So it's more around concept like communication, healthcare staff relationship, adherence to treatment. So the concepts are a bit different. Here is just an example of a PREMs, so the consultation and relation and empathy, the care questionnaire. So now that we know a bit more about what is a PROMs and what are PROMs and PREMs, how they are structured, what it means to develop a questionnaire, now what I should consider before including them in a trial. Just the first major thing is to know what you want to measure with such tool, because not one questionnaire can fit all the needs. So it's very important at the very, very beginning to clarify what is the objective of your PREMs or PROMs. So what do you want to measure? There are different types of, let's say, big category of assessment. So first, with the PROMs or PREMs, you want to measure a disease burden on patient quality of life. The second could be to assess quality of care, communication with healthcare staff, or to support the medical decision-making process. Or also to evaluate a treatment, but it could be a surgery, could be any kind of intervention, could be also a device. So first, that three big categories, let's say. But it's really important to define this at the very beginning, just to be sure that you're not adding a burden to the patient because you don't really know why you're using it, but someone told you that you should use it, but you don't really know why. So the first step is really to know why I'm using it, just to be sure that it's worth using a questionnaire. The second aspect, beside the objective, is to really understand the context of use in which you will use this questionnaire. Because as you can see, depending on the context of use, maybe the questionnaire itself will be different. So here is just an example. So here it's the measure of depression, and we'll see 4 different context of uses. So the first one is the screening. You want to screen, so you have a patient and you would like to screen and assess if this patient is depressed. So the appropriate scale will be the Hospital Anxiety and Depression Scale, the HADS, which is a gold standard for assessing depression. It's 14 items, so pretty quick in terms of time of completion. And you get an immediate score, allowing you to assess if yes or no, or the different level of depression levels. Then if you want to measure the impact of depression on the quality of life of patients, here you have different type of scales with different numbers of items. So here it's more a one-shot assessment or one or twice, because as you can see, the first two, we have 34 item or 36, which could be pretty long. And meaning a bit of burden for the patient if they have to assess it like every week, for example. Another aspect is here to assess the midterm outcome of a care in all patients in a hospital. So it's more like a cohort or the assessment of a performance in an hospital, or like to extend um, the assessment of depression in general. So here we suggest the motivation and energy inventory, 18 items. And the last one, to reflect the assessment of a drug treatment or an intervention treatment, here is the Montgomery-Åsberg depression rating scales with 10 items, really focusing on sign and symptoms. So in a nutshell, what to consider before including PROMs or PREMs in a trial or in your daily practice. First is to define your objective and what you want to measure with this tool. Then you can look for PREMs. So you will see the next part is where I can look for suitable PROMs or PREMs. Then you will end up with a list of different PROMs and PREMs. Then based on the condition of use, you can narrow down this selection to what you want to measure, fitting your objective and condition of use. And based on this short list of questionnaires, then you can identify further validation steps. Should I take this questionnaire, adapt it to my population, or should I develop a new questionnaire? That could be also an option. This last part is more in terms of beyond the validity of the PREMs. Even if you pick up the questionnaire that has followed all the validation and development steps, so they should fit all the requirements in terms of statistic, you need to make sure that the questionnaire is accepted by both the patient and the clinical staff. So here are just 4 criteria that will turn your questionnaire, to consider to favor the acceptance of this questionnaire by both patient and clinician. First, to make sure that the questionnaire is easy to use, it's feasible. So here it's, for example, in terms of response option, you can have a numerical rating scale or a visual analog scale. And numerical rating scale, it's easier to rate and score. So it's easy to use compared to the VAS. Is actionable. It's what we just talked before about the importance that the score of the PREMs or the PROM is easy to get and easy to interpret to lead the decision-making process. You need to make sure that the PROMs is appropriate to the context of use, as we have just said before. For example, here to make sure that it could be incorporated into the provider routine process of care. The SF36 here is a good example, 36 items. It's quite a long list of items. It's very good for research because it allows a comparison with many patients, many diseases, but it's a complex scoring and doesn't allow like a routine use. And obvious, but still it's important to mention it, that the questionnaire should be useful. It needs to contribute to better healthcare and not used because it's kind of trendy. So at what point I need to think about PROMs? So here I just wanted to share this because it's super detailed, it's super exhaustive and can really guide you. We're not going into many details, but it's just to show you the three main aspects on what to consider when you are in using PROMs or PREMs. It's basically also a recap of what we just discussed before. But it's just another way to visualize it. So the first thing is really to better understand the disease or the condition. It's a very crucial, important step before even thinking about the tool itself, is to know the natural history, to know your patient population, the healthcare environment, and also the patient and caregiver perspective. We know that the caregiver, it's an important perspective to get also before including a questionnaire. Then it's conceptualize the treatment benefits. So to understand the concept of interest that are meaningful in terms of treatment benefit, the condition of use for the clinical trial, and to select the appropriate type of COA, the 4 that we have just seen before. And then, as you have completed the 2 previous steps, then you can look for existing questionnaire appropriate to measure your concept of interest in your condition of use. Maybe begin a COA development and complete a COA development. So the last part is where you can find suitable PROMs and PREMs, and how I can, if I have question, who I can contact to get more information. So in the work packet 3, as a background, we have, so here is just to show you the gap analysis that we have performed last year based on the different therapeutic area and the number of questionnaires that fit that have been developed for a specific therapeutic area. And roughly the conclusion is like we can see that for some therapeutic area there is no questionnaire, and for others, here in neurologic diseases, for example, there are way more questionnaire developed for the therapeutic area. So we end up with this conclusion that there is a huge heterogeneity in terms of PROMs that are available for specific diseases. So that's why we have decided to develop a repository that is a tool where you can find interesting or suitable PROMs. And for that, we perform a PROM selection to include these PROMs into this repository. So we have, as a first version of this repository, there is a little bit less than 300 PROMs specifically developed in rare diseases. The gold standard generic questionnaire measuring quality of life, PROMs measuring specific functional impact. So they are not necessarily developed for a specific disease, but they are developed for a specific functional impact, like understanding, mobility, activity of daily living, social functioning, etc. And also questionnaire that assess healthcare management concept, like adherence to treatment, treatment burden, family impact, communication with healthcare staff. And we also ask ERN members to get their feedback on the questionnaire that will be interesting to see in the repository. So we end up with a final list of 811 PROMs. That's the first release. I'm going to see you in the next slide a screenshot of the repository. It's free of access, so you can all access it. And there will be a release very soon, next month, including ops row to reflect the need for pediatric population. And also we perform a wide review of the most recently PROMs developed for rare diseases, developed and validated for rare diseases, to increase the number of PROMs developed in rare diseases. So here is the screenshot of the repository. You can look through. I mean, there is a advanced search engine where you can look through the type of questionnaire that you're interested in, the name of the questionnaire if you already know it, the age range if it's adult, pediatric, or older people, the disease name, the group of diseases, and also by ERN. You can see at the very end of the table that there is a link to another database, which is Mapi Research Trust database called PROCOLID. And here it's another step where you can find for each questionnaire described in this repository of a more complete set of information on the author information, the condition of use, the available translations, the population for which it has been developed, the review copy of the questionnaire, and also information on the development and the validation. For your information, the frame of ERICA project, all ERICA members has a free access of this database. So please, if you're interested in getting access to this database, ProColleague, then send an email to Marie-Muriel.
Well, thank you very much. I'll take over from here. My name is Andreas Glenthøj. I'm a medical doctor and I head the Danish Red Blood Cell Center here in Copenhagen. We treat and diagnose patients with rare diseases. As the name implies, we treat red blood cell diseases. We run many different trials. Most of them are pharmaceutical companies that initiate trials, phase 1, 2, 3 trials. But what is interesting in rare diseases are often investigator-initiated trials. And in this context, I would like to introduce the SATISFY trial, which is a phase 2 trial that I've run together with academic colleagues in the Netherlands and in Canada. We have an intervention, which are tablets, mitapivat, which are approved for another indication by the FDA and by EMA. What we want to do is to treat it for some other rare entities, hereditary spherocytosis, hereditary stomatocytosis, and congenital dyserythropoietic anemia type 2. And where the first one, spherocytosis, is not that rare, the other two are ultra rare, especially the last one. So why use patient-reported outcome measures in these diseases and in rare diseases in general? Many years ago, I would probably have been able to conduct a clinical study just saying, well, these patients have anemia, they have a low hemoglobin. I do an intervention. If the hemoglobin goes up, it's likely that the patient will benefit and the drug might be used for that indication because of the trial that I do. But history is filled with different examples of these paraclinical measures that actually do not reflect that the patients benefit from the treatment. So luckily, nowadays, we also have to prove that the patients actually feel better. And that's where these patient-reported outcome measures come in. But it can be difficult, as mentioned before. Often we don't have disease-specific patient-reported outcome measures for these ultra-rare diseases. And also, it takes a lot of time and effort to manually do them. And for my small trial, it would be very, very difficult to develop a specific one for these diseases. So another option is to take some that are similar patient-reported outcome measures and adapt them to these diseases. And as you see later, we've done that instead. But we also have to look at some parameters for choosing the right patient-reported outcome measures for this specific trial that we did. First of all, I don't have a lot of resources, and it's very burdensome to have a paper that needs to be typed in. So for our purpose, at least, it was much easier if we could find one that had an electronic version or that could be adapted to an electronic version. We use the REDCap database system, which supports sending automatically these surveys to the patients. That makes it easier to manage, and we can do different reminders if the patients, they don't type in the patient-reported outcome measure surveys. Also, the patients can type it in the waiting room, or they can get an iPad when seeing me and type directly in. Obviously, if the patients are not comfortable using electronic devices, they can get a printed version instead that we can put into the database. So you don't need to use the e-version, but it can be much easier if the patient group actually is one that you would expect being comfortable with electronic devices. Secondly, we run the trial in Canada, in Holland, and in Denmark. So we have 3 different languages that we need to use. It's a lot of hassle to translate these tools into different languages. You need to translate them, and you need to translate them back again to make sure, 100% sure, that you're actually asking the same questions in different languages. So the easy way to go around this is obviously to find some that are already translated and back translated and thus validated in different languages. And we succeeded in finding that as well. Then you need to look at the patient age. So if you're studying pediatric populations, you need to have some tools that are understandable and applicable to that age group. We run an adult trial for 18 plus, so it's not really a problem here. The tools are suitable for that. Then there's the last part. It can be expensive. Some are freely available or some are free to use. Others cost a lot of money. And in a small trial for a rare indication where we don't have a lot of money, we need to take that into consideration. So if we take the two examples we used, there was the SF36 that was mentioned previously. That's a pretty widely utilized patient-reported outcome measure. It was done many, many years ago, and it's already available in an e-version in our REDCap database. So I could just pull it in without any real effort. And even the scoring system was pulled into that. The translations, they were available, not in the REDCap database, but online in paper version. So it was fairly easy just to do the translation. It didn't take a lot of time, and it was still fairly easy to send to the patients. It was also validated for adults. That was easy. And then there was the problem about the licensing fee, because in this case, at least, version 1, which has one scoring system, that's freely available. You can use it for anything you want without paying anything for it. That's quite attractive. And then there's a version 2.0, which has its advantages, definitely, but there's a lot of fees and it can be burdensome. There was some response time from the company we approached. There were some costs and there were also some conditions on how we could use it. So that made it difficult. And for that reason, we chose version 1. Then we also needed to have something that at least looked a bit more disease specific. And in this case, we knew that there was a tool, we used it in commercial studies, called the PKDIA, the pyruvate kinase deficiency impact assessment, which is used for another, at least phenotypically similar anemia. It was used in a commercial setup. We needed to type it into the REDCap database. That didn't take a lot of time. And it was available from the company who did it. And they were interested in us utilizing it. So that was good. It was also validated for adults, and it was translated into all the languages we needed already. So obviously, the licensing fee was, for us, nothing. But that depends on which use you had. And obviously, a commercial company owning that would need to accept it being used in the specific study. So with that, that was my input, and thank you very much.
Hi, everyone.
Hi, there's one of the questions here. I can see if we can reduce drug development timelines and help in addressing high costs of certain orphan drugs. I think that's a very important question. One of the reasons why we included PROMs in our study was obviously that we not only needed to improve the hemoglobin, but also show a real benefit of patients. And I think it's essential to prove that part. And without those data, we won't get the drugs to the patients. So if the question is the development timeline is from development of the drug to it actually reaches the patients as an approved commercial drug. No doubt about the importance of PROMs. There's no way around it. And with the very high cost that some of these products have, I think it needs to be done. It's not that expensive compared to all the other things we do biochemically. It's fairly cheap. So yes, I think it's important. Any other comments from my fellow persons on stage?
Nope. That's fine for me.
I'm not aware of translation service we have for questionnaires. That's another question. Obviously, Céline, you're probably better at that.
So for the translation, that's a good question indeed, because it could be a major barrier in terms of timelines or even in terms of skills to translate. So one thing will be, so in the PROCOLID databases, you will have access to all the existing translations, the one that we have done internally, but also the one that has been published. So it could be a first good step to check in the specific questionnaire the available translation. And otherwise, if you need a translation, you can submit a request under this database referring to ERICA, and then we can see how we can enter the request. When ICC, it's like in terms of fees, deadline, how it can be supported within the frame of ERICA. So really feel free to check first in PROCOLID, then submit a request or send me an email, and then we can see what we can do in terms of translation. Okay, I'm going through the rest. Is that, yeah, did I address the question? Okay, then I'm checking the other questions on. So the question from Sanjita, so about the drug development. Oh no, it's not this one, sorry.
There's a question at the bottom I think is very good. How effectively can PROMs be used as real-world evidence to design safety and efficacy studies framework for orphan drugs? I think that's an excellent question. Actually, I am aiming to, for instance, in one of the patients population I study, to do a PROM study of the patients we follow with these rare anemias in different centers around the world, to study the burden of disease, to sort of less evaluate where do we see room for improvement, what are the patients affected by. So I think it can be used to generate evidence before doing a real clinical trial. That's definitely worth the effort, especially if you want to prove an effect of a drug on a PROMs. It's very good to know that it's actually the patients score differently than the general population on these PROMs. Otherwise, obviously, you won't find any difference from the drug.
If we want to move forward to your question on sickle cell drug development, and with the two example of yes, this one, thank you, for for the disease.
Yeah, I think when, for instance, when Oxbryta was presented, they used a pseudo measure of hemoglobin improvement. And to be honest, that has been used before. That was used many years ago in the Zanikapok trials, where we saw that hemoglobin increased in patients with sickle cell anemia when getting this form of drug. The problem was that the patients got more vaso-occlusive crisis, and the phase 3 study failed. So that's an excellent example of a biochemical measure being actually harmful for the patients. So in this case, when Oxbryta was presented, the hemoglobin increased, but I think a lot of people were quite skeptical that it actually improved the quality of life of the patients. But later on, they presented PROMs data. I don't have all the details. I'm sure many of the audience know them better than I do, but it actually showed improvement, and now the drug is being somewhat utilized. It's more a pricing issue, I guess, how many patients get the drug.
Yes, and so far, none of those two, let's say, chemical has been approved with PRO data in the label of drugs, either from EMA or from FDA. For the moment, the measure that has been recognized in the endpoint strategy of the trial are more focusing on clean rows. It's what we got from our drugs database.
But it's safe to say that to get not just conditional approval, but final approval for a drug, you would need to present data like this. And in the case of crizanlizumab, I think many in the audience are well aware that the regulatory authorities are looking into to this drug in particular. But it just stresses we need to include it. But obviously, often the biochemical data is the first to be presented, and later on this is being presented. But if the data is never recorded, you won't get that out of it. It will. If we look at the gene trials, the Bluebird gene trials, they didn't include PROs or PROMs in their first patients in the gene therapy trials, and that was a mistake. They corrected that later on, but obviously that would have been a major issue in getting approval for the drug, which unfortunately is not available in Europe, as many are aware. I think that's what I had, at least, for that question. We have two left. The translation, Céline, you mentioned that?
Yes, yeah.
So what is the process where you need to develop a new questionnaire for your rare disease, since the ones available are not suitable? Who does the final validation?
Okay.
You're probably the expert there, Céline.
Yeah. Okay, so the process, I think there was one slide on the process to if you want or need to develop questionnaires. The first important thing is to document the disease, to be sure that you fully know, you're fully aware of the impact of the disease. And then go through this round of patient interviews, then patient understanding, then the first round of statistical analysis to make sure that what you want to measure, you can actually measure it with a questionnaire. There is no redundancy. And then you go through the final set of validation statistical analysis. So this is the process. You will have access to the PowerPoint, so you will see very detailed. And it's also depending on what is the intended use for the questionnaire. If it's for drug approval process or if it's for clinical practice, you can make some adjustment. And who does the final validation? So usually the final validation is statistical analysis. So it could be done by specialists in stats. So either researcher or either company like ours is doing. We are expert in developing and validate questionnaires. So it's depending on resources. So the same, if you have a specific questionnaire in mind, maybe it's worth within the frame of ERICA project to talk with me just to see what is out there, what is available, and how we can help the work package 3.
So from my count, that was the questions asked there. And there's also a few nice words, compliments on the chat. So thank you very much for that.
Yeah, thank you. And you will receive your our contact. So if you have more specific questions afterwards, really feel free to send us an email, and we'll be very happy to address them within the context of ERICA project or other context. Thank you, Simon.
Terrific.
Okay, maybe we could wrap up here. Giacomo, I don't know if you confirm. Yes. There will be news also with the workshop. The work package 3 is preparing at the end of May, and also during the General Assembly that we will have at the beginning of July.
Terrific. Well, I guess we can wrap this up. Thank you very much for attending.
Yeah, thank you all.
Yes. Thank you.